Now we'd like to start the session 2, latest trends in regenerative medicine. I would like to invite Dr. Masaya Nakamura, Vice Dean of Keio University School of Medicine and Professor and Chair of Department of Orthopedic Surgery.
Good evening, everybody. It's my great pleasure to be here to present our recent study about regenerative medicine for spinal cord injury using iPS cells. Okay, let me begin my talk with this slide. As you know, this is Lamoni Kahar. He said, spinal cord injury results in devastating loss of function because spinal cord never regenerates after injury. This is what is called Kahar's dogma. People believed in this dogma for a long time. In fact, this is my patient. He was 18 years old football player. During the game, he suffered from complete tetraplegia due to the fracture dislocation at C5-6 level, as you can see here. In order to decompress the contured spinal cord and reconstruct the spinal column, I performed anterior spinal fusion with plating and posterior wiring. This is post-operative X-ray films. It looks pretty good. However, his tetraplegia never recovered after injury, as you can see here on this MR image. Here. There are huge cavities and glial formations, and the spinal cord was completely ruptured. Can you imagine how devastating his situation is? All he can do is just moving his shoulder like this way. That's it. No other movement at all. This is a reality we are facing in clinic. This is the background. However, recent progress in neuroscience has opened up a possibility to cure the patient with spinal cord injury. Challenge towards regenerating damaged spinal cord is quite similar to the huge jigsaw puzzles. I mean, we have to put together a lot of different pieces, such as cell transplantation, overcome axonal growth inhibitors, rehabilitation, imaging, growth factors, and scaffold. I'm pretty sure there still exists a missing part of these jigsaws. In my today's talk, I focus on cell transplantation, especially iPS cell-derived neural stem cells, iPS-NSC. In tight collaboration with Professor Yamanaka and Professor Okano, we performed several preclinical studies of regenerative medicine for spinal cord injury using iPS cells. There are two main topics today. One is subacute SCI, the other is chronic SCI. First topic is subacute SCI. For the sake of time, I just focus on the study using non-human primates. This is a picture of common marmoset, which we use in our studies. This picture is a transplantation site. Actually, we induced severe contusion injury at the C5 level. And 2 weeks later, we performed the transplantation of human iPS-NSC into the regional center. The grafted cell will survive at the regional center, and they differentiate into neurons, astrocytes, and oligodendrocytes, resulting in this is a transplantation group, this is a control group, resulting in the increase of the areas of L3 positive area as well as the communicated positive areas. This is a marker for corticospinal fibers. And eventually, we performed behavioral analysis by original open field scoring scale. And we found that there were significant difference in this scoring scale between these two groups. at 56 days and thereafter. But it's kind of difficult for you to understand how their performance are different. And I'm going to show the movies of the representative animals in each group. This is the control animals. Because we induce severe cervical contusion injury, they suffered from severe tetraplegia. No matter how hard we push them to move, they could not move forward by themselves. We tried to push them to move, but they can't. Next is bar grip test. Okay, they could grasp the pen perpendicular to their body plane at the eye level, but they could not grasp the pen parallel to their body plane because their forearms were not pronated enough. And they could not reach the pen 3 centimeters above their head because they could not elevate their shoulders. Almost better, basically very close, but they couldn't make it. And next is the cage climbing test. We put the marmoset at the side wall of the cage upside down. Due to severe tetraplegia, they could not keep their body by themselves. Okay, try again. They could not support their weight by themselves. They were almost holding down. This is the control animals. Next, I'm going to show you the transplanted animals. You can see a difference at a glance. They can walk around. Just a break, but they could walk around. Next bar grip test. They could grasp a pen very quickly at the eye level, as well as they could reach a pen 3 centimeters above their head because they could elevate their shoulders. See? Okay, they could make it. And surprisingly, they could keep their bodies by themselves and change direction and climb up the cage. You can see a difference between these two groups. These are potential mechanisms of functional recovery after iPSC transplantation. One is synapse formation by graft-derived neurons. Another is remyelination by graft-derived oligodendrocytes. The other is trophic support. Actually, iPSC secrete several growth factors such as BDNF and or NT3s. Let me explain a little more about these two mechanisms. Triple immunostaining with these markers suggested that mouse-specific bad zone co-localized graft-derived neurons, like here, and also the human-specific synapse fusion co-localized host mouse neurons here and here. Furthermore, immuno-EM examination revealed that there were post-synaptic densities here, between the graft-derived neurons and host neurons. All of these findings suggest synapse formation between the graft-derived neurons and host neurons. We call this relay mechanism. Okay. And next is remyelination by the graft-derived oligodendrocyte. This is a transplantation site, and this is a control group. As you can see here, there are grafted cells at the regional epicenter. And you can see a difference in the LFB positive myelination area between these two groups. And triple immunostaining revealed that the GFP positive grafted cells differentiated into MVP positive mature oligodendrocytes and contributed remyelination of the demyelinated axons. Okay, this is our original strategy of regenerative medicine for spinal cord injury using iPS cells. We planned to establish the patient-specific iPS-derived neural stem cells and transplant them into the spinal cord, injured spinal cord of the patient. That's our original strategy. However, there are several concerns. One is therapeutic time windows. Actually, it takes about 6 months to obtain patient-specific iPS-NSC, which means we miss the golden therapeutic time windows for subacute SCI patients. Another concern is quality control of iPS cells. Actually, to overcome these issues, iPS cell stock was established by Professor Yamanaka to obtain iPS cell stocks for clinical use. But our most serious concern was tumorigenicity. To overcome this issue, we performed in vitro as well as in vivo evaluations of iPS cells and iPS-NSCs for over 5 years. We spent a lot of time and money to overcome this issue. And eventually, we established the protocol of iPS-NSC production. These are the examined items. Sorry, it's too busy, but for the sake of time, I want to skip them. This is a roadmap to the clinical trial of iPS-NSC transplantation for subacute SCI. Clinical grade iPS cells were established at Kyoto University at Saira. And through the in vitro evaluations of iPS cells, they selected a good clone of iPS cells and sent them to us. We induced neural differentiation, and through the in vitro as well as in vivo evaluations of iPS-NSC, we select the best clone of iPS-NSC for clinical trial. And before starting clinical trial, our proposal had been deliberated by Scientific Council of Japanese government, which was accepted at February 10th, 2019, almost 3 years ago. This is outline of this clinical trial. This is an interventional open-label clinical trial, single loop assignment, and we plan to recruit 4 patients with AGA grade A, complete spinal cord injury, at 2 to 4 weeks after the spinal cord injury, whose age is over 18 years old. We plan to perform transplantation of human iPS-derived neural stem cells into the injured spinal cord through the intraregional injection. And primary outcome measures are types and frequencies of adverse events and increase of AGA motor score from the baseline at 6 and 12 months after transplantation. There are 2 important points of this clinical trial. One is neurological status. These bar graphs, line graphs, show the changes in the AGA motor score of the patient with spinal cord injury at the Spinal Injury Center at Fukuoka Prefecture, about 982 patients. This is the averages of the changes in the AGA motor scores of the patient with AGA grade A and B. At the beginning, we plan to recruit the patient with AGA grade A and B, which means motor complete injury. But it turned out there are significant difference in the change of the AGA motor scores from the baseline between grade A and B, here and here. That's why we decided to recruit AGA grade A patients only in our study. No B. Of course, no C. Maybe you don't know. The patient with AGA C, about 40% of patients with AGA C can work. Around 40% of the patient can work without any treatment. That's the reality. That's why we excluded this kind of incomplete spinal cord injury from this study. The second point is patient age. Over 18 years old. No upper limitation. That's the point. We try to determine whether we should consider the patient with take patient age as inclusion or exclusion criteria. We performed the next two experiments. One is we compared function recovery after moderate HCI between young and aged mice. Actually, this is 10 weeks, yes, 10 weeks age, and aged mice is about 60 months old, very old. And behavioral analysis revealed that the aged mice showed, the HAPA is a better function in terms of function recovery. The aged mice showed less function recovery after moderate HCI than that of the young mice. Okay, this is the baseline. And next, we compare the functional recovery after NSC transplantation in moderate HCI model between young and aged mice, and these whole groups. And behavioral analysis revealed the aged mice showed dramatic improvement here in BMS score after transplantation comparable to those of the young mice. We are so surprised with this result. This result prompted us to examine microenvironment of the injured spinal cord. Okay? And the comparison of gene expression in the injured spinal cord between young and aged mice revealed that endogenous HGF gene expression is significantly higher in the aged mice compared to the young mice. Therefore, we examined the effect of HGF functional blocking on differentiation of the grafted cells and function recovery in the aged mice. And here you can see HGF functional blocking hindered neuronal differentiation of the grafted cells here and the motor function recovery after NSC transplantation. Okay, this is a summary. And all of these findings suggested that aged mice with SCI exhibited enhanced regenerative capability following transplantation, even though they suffered more severely from the SCI than do the young mice. Based on these results, we decided not to exclude the aged patients with SCI from our study. Okay, this is the workflow of a clinical trial for subacute SCI patient. We obtained iPSL from CIRA and induced their differentiation into the iPS-NSC. And after the in vitro as well as the in vivo evaluations, we make a freeze stocks of iPS-NSC. Actually, we completed these evaluations May 2020, almost 2 years ago. However, we could not start this clinical trial due to the pandemic of COVID-19. It was so stressful. We started the recruitment of the patient, but postponed it, and restart the recruitment and postpone it again. Eventually, we restart the patient recruitment October 28th to 2021. And finally, we performed first in human transplantation of iPS-NSC for subacute HCI. This is the preoperative MRI of this patient. You can see a huge T2 high signal intensity area and a T1 low signal intensity area. The spinal cord almost ruptured. This is the intraoperative videos. After exposure of the spinal cord under the microscope, I injected 2 million of iPS-NSC into the region cavity manually. And you can see that I was so surprised. The surface of the spinal cord looks pretty good. No vascular damage, no adhesion. I was so surprised. But inside at the regional episode, there are huge cavities. And so far, I performed the iPS-NSP transplantation on the patient with age A, 2 patients so far. Hopefully, I will be able to make a presentation of the result of this clinical trial next year in detail. This is the story about subacute SCI. Okay, next topic is chronic SCI. Oh, my timing is running out. It's okay. Okay, a little bit speed up. Okay, actually, in the subacute clinical trial, we used 4 brain type of iPSNSC. This is a heat map of the gene expression of these cells. But from the viewpoint of developmental biology, spinal cord type of iPSNSC is more suitable compared to the 4 brain type of iPSNSC in terms of the regenerative capability. That's why we developed original differentiation protocol of spinal cord type of iPSNSC and use these cells in the chronic SCI models. Actually, we induced severe contusion injury at the thoracic levels, and 18 weeks after the injury, you can see here severe spinal cord atrophy and cystic cavity formation were observed at the region epicenter. However, maybe you will be surprised with this. The spinal cord atrophy was completely attenuated because grafted cells will survive at the regional epicenter, and these cells migrated into the rostral caudal to the regional epicenter. Furthermore, see here is the regional epicenter. The spinal cord tissue almost completely replaced by the grafted cells. And more interestingly, grafted cells migrate into the dorsal column of the caudal spinal cord. Here, cortical spinal tract was distributed here. Oral generation occurred here. That's why they migrate into this area. Furthermore, the grafted cell differentiate into the neurons at the regional epicenter, CHAT positive cells, Hu positive cells, and the grafted cells differentiate into oligodendrocyte to the caudal site. And this is a picture after phosphorylated GAP43 staining. This is a marker for regenerating axons. Their number of phosphorylated GAP40 fibers even after clonically transplantation. And interestingly, these GAP43 positive fibers form the cluster here and here. And the quantitative analysis of GAP43 positive fibers area revealed that there were significant difference in this area between the control and the transplantation groups. And these findings were also confirmed by MR imaging technique, okay, which is diffusion tensor tractography. Actually, this image was taken at the Central Institute of Experimental Animals, Jichuken. And this is depicted fibers of the spinal cord pre-injury. After injury, before transplantation, these depicted fibers were significantly attenuated. The very few fibers were depicted. But after transplantation, surprisingly, you can see here a number of fibers were depicted even at the region epicenter. And eventually, this is a control rat, and this is a transplantation rat. You can see a difference. Grafted animals could weight their body, bear their weight on their hind limbs. And this is a BBB scoring scale. There are significant difference between these groups. Actually, based on these results, we are planning to start the clinical trial for the chronic SCI patient of transplantation of spinal cord type of iPSNSC, maybe within a couple of years. This is the status of the chronic SCI. And I'd like to express my sincere gratitude to all members, especially Professor Okana and Professor Yamanaka and Dr. Nomura. Thank you for your listening.
Thank you, Dr. Nakamura. Now, as the next speaker, I would like to invite Dr. Peter Kao, Associate Professor of Medicine, Pulmonary and Critical Care Medicine, Stanford University School of Medicine. The title of the presentation is ILF2 and F45 regulates cell proliferation and pluripotency, and it represents a novel therapeutic target in cancer.
My gratitude to the organizers for inviting me. It's a pleasure to be here and to share ideas and stimulation. So this work goes back 30 years, and this is work I did as a postdoctoral fellow at Stanford. And I discovered a nuclear factor, one of a pair, and I'm just going to talk about the small factor that is so important in regulating pluripotency and has been identified by other groups as being highly expressed in embryonic stem cells, driving proliferation over differentiation, and it also is upregulated in cancer and confers stemness to cancer cells and therefore can be a very important therapeutic target. So honoring Professor Shinya Yamanaka, who very elegantly identified a set of 4 reprogramming factors. These factors themselves are proteins that exist in the nucleus, and they are transcription factors. They drive the expression of target genes, and the Yamanaka factors maintain pluripotency and can reverse differentiated cells into a phenotype of embryonic stem cell-like or induced pluripotent stem cells that can then be differentiated, as we so elegantly heard, to be used for therapeutic purposes. So the factors that I discovered, and this is work of Guha Zhao and F45, it's actually expressed in most tissues, largely in the nucleus. These are hippocampal neurons. These are cortical neurons. These are Purkinje cells. This is in the sperm, highly expressed in the sperm, in the spermatocytes, but not in the mature sperm, expressed in the liver, expressed in the kidney tubules, but not the glomeruli, expressed in bronchial epithelial cells, highly expressed in islets of the pancreas, and contributing to the capability of beta cells to make insulin, expressed in muscle cells in the nuclei. They're located primarily in the nucleus. They regulate cell cycle. And the way I originally undertook to study these was I went to a T-cell laboratory at Stanford after I'd witnessed the AIDS epidemic in New York City, and we were purifying nuclear proteins that could regulate the transcription of interleukin-2, an inflammatory cytokine. And this shows that if you overexpress these proteins in Jurkat T cells, you can 50-fold increase transcriptional activation, so they are functional transcription factors, and you can increase the expression of IL-2 protein, so they are cytokine regulators. And if you diminish the expression by using a reverse cDNA, you can attenuate the expression. So this is work by Yosuke Aoki, who's a professor of infectious disease at Saga Medical Center, my first postdoc, and he's going to come visit me at Haneda Airport on Saturday so we can get together after 25 years. And the staining shows that these are expressed in the nuclei of cells. They also show here that these proteins move into the cytoplasm during mitosis, that's cell division. And so this was our first suggestion that they contribute to the regulation of cell cycle. This is work much later of Atlanta Cook, a crystallographer in England, that co-crystallized these proteins together and identified that there are conserved dimer of zinc finger domains in the small subunit NN45 and NF90, and they dimerize across antiparallel alpha helices here. We have thought for 25 years that they always work as partners, but they actually breathe. So they come together and they come apart. They are translocated from the cytoplasm into the nucleus on the back of NF90, which has a nuclear localization signal. And the proteins themselves are predominantly RNA-binding proteins that have RG-rich domains here, and also double-stranded RNA-binding domains. You'll remember that the Cas9 protein that Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for CRISPR-Cas9, the Cas9 protein holds on to RNA through some double-stranded RNA-binding domains and then has a free end that directs Cas9 to different places in the genome. And I wonder whether these proteins are chromatin targeted by RNAs that are being slipped in here, and whether that's why our chromatin immune precipitation shows different chromatin locations in different cell types. So this is work of others that have informed my understanding of how these proteins work. Mike Matthews at New Jersey showed that the NF45 subunit turns over very rapidly with a half-life of only 1.5 hours. So it's always being synthesized and always being degraded by ubiquitin-mediated degradation. And this steady-state level, that's the balance between synthesis and degradation, informs how much of the ILF2 protein is present in a cell and roughly drives cell cycle and drives cell proliferation. If you knock down NF45 with RNA interference, you end up getting multinucleated giant cells, which are defective in mitosis. There's a failure of terminal cell division called cytokinesis. That's shown here in work from 2020. This paper came out, and they also knocked down NF45 or NF90 and showed defects in cytokinesis and mitotic catastrophes. So this suggests that if you knock down the expression of one or the other of these subunits, you can arrest cell cycle. There was work by another set of laboratories. Dr. Blaylock is at UCSF, and Julia Ye was an MD PhD student, and she characterized the importance of ILF2 and ILF3 as pluripotency factors in embryonic stem cells. This is work from this laboratory, Dr. Hori's lab, that actually made knockouts in embryonic stem cells. This is published in 2011, and the ILF2 knockout has much smaller spheres and a longer cell doubling time of 14 hours compared to 10. If you revert this, you can restore the doubling time. So clearly the levels of NF45 inform the doubling time. And this is a network of pluripotency factors in embryonic stem cells. NANOG here is one of the central factors, OCT4 here, one of the Yamanaka factors, and here is ILF2 and NF45, identified independently as one of the network of factors in embryonic stem cells that drives self-renewal and proliferation. And finishing up, it turns out that a number of different cancers are characterized by an overexpression of NF45 or NF90 or both. And this is an example of esophageal cancer showing immunostaining, where heavy smokers have very strong nuclear expression of ILF2, and it's much higher, statistically higher. And if you knock down the expression of ILF2 by RNA interference, xenografted tumors in immunodeficient mice, this could be NOG mice, grow much less well, identifying ILF2 as a gene target that can regulate tumor growth. And this is a set of work by these investigators that showed that pluripotency factors in cancer cells, SOX2 and NANOG and SAL4, are upregulated when you overexpress ILF2 and downregulated when you inhibit ILF2. So the conclusion from Lee et al. Was that ILF2 is induced by nicotine in esophageal cancer cells. It stabilizes the pluripotency factors in cancer cells, contributing to cancer stemness and chemoresistance. So this work identifies ILF2 as a novel therapeutic target in cancer and a useful therapeutic target to help promote the regeneration of cells. So thank you.
Thank you, Dr. Keo. Now, I would like to introduce Mr. Nomura for the next presentation on the regenerative medicine.
So I thought that I make some comment during the discussion, but I was mentioned. So I like to talk about CIA's activities in regenerative medicines in Tonomachi. So CIA is a non-profit institute for the especially in vivo studies. So what in vivo study our non-human primate super immunodeficient nog mice is contribute to the regenerative medicines. One is CIA, the co-development with many Japanese famous professors, such as Professor Nakamura and Professor Okano of Keio University for spinal cord injuries, and Professor Jun Takahashi, who is now the head of SILA for the Parkinson disease, and the Professor Masayo Takahashi is the Riken and the now Vision Care president for the retainer regenerative medicines. And on top of that, our animals is used for the safety study for the SILA's Professor Yamanaka's study, especially for Professor Nakamura mentioned that SILA has the iPS bank. So our NOG mice made a study for all these iPS cells banked in SILAs. So on top of that, the CIA contribute for the in Tonomachi's the value chain for the regenerative medicines safety study, together with NIHS and Riken and the KISTEC in Tonomachi. And we have so many variety of the companies and the institute who make a support for the regenerative medicines research. That is most the strong point of King Skyfront Tonomachi. So that I like to show what King Skyfront contribute for the regenerative medicines research. Thank you very much.
Thank you, Mr. Nomura.
Okay. Thank you for your three excellent presentation. We have time limitation, next only 5 to 6 minutes. By the way, we have distinguished panelists here from Taiwan and also industry field, Dr. Yamada and Dr. Chen Yen Ko. Maybe we have only 5 minutes. Therefore, let me skip your introduction. However, from the point of view, gene editing side, I'd like to comment from Dr. Yamada, as well later on, Chen from the point of view or economic or finance or business point of view. Please give us your comment on this regenerative medicine, Dr. Yamada.
Thank you very much, Nishimura Sensei. And before my comment, so Paru Sensei, Kao Sensei, Nishimura Sensei, thank you very much for your giving us a good opportunity to have an agreement with you, Stanford University, about the COVID-19, in a sense, nasal injection technology and so on. And as Nishimura Sensei mentioned about the gene editing area, we merged 2 years ago American company Emend Bio, and that company is focusing on the gene editing area. Their technology is different from CRISPR-Cas9, and in the case of CRISPR-Cas9 has some kind of problem about the off-target matter. So in the case of Omni technology derived from Emend Bio, in that case, so off-target is extremely decreased. And later, this week, Friday, I will meet with you about talking about this matter. So anyway, off-target is to be a good progress, decreasing a good progress OS. And furthermore, in the case of this technology is transfer vehicle, for example, LNP or AAV or antivirus is a very key virus vector. So we are examining the situation of each indication, and case by case, the selection is to be reasonable considered at this time. So anyway, in this area, as you know, from the viewpoint of business, so United States is very excited. And in the case of gene editing company, almost IPO company has more than $1 billion US dollar market cap. Of course, bigger market cap makes big R&D budget. It's a different from Japanese circumstances, so it's a big problem for us. So if there's not a change of this situation, so in the future we have to move to United States area, so it's a problem for us. Anyway, so this Friday, so I will explain the current situation of this gene editing areas derived by Emend Bio. So anyways, thank you very much, and I'm very excited to...
Please stay in Kanagawa.
Yes.
And Chen,
yes.
Chen, please, your comment.
Yes.
Yes. Okay. Thank you. Yes, I'm not a scientist in this field, so I cannot comment on scientific part. But I was so amazed by the three speakers that the progress of regenerative science has made. Especially, it's not just in the research lab, but also as it's being performed by Dr. Nakamura. So I think I agree that but I think in general, as an ordinary person, I may not have opportunity, if I did not come here today, may not have an opportunity to understand the progress of this science and its potential application and benefit to the societies. So my suggestion is this is really for the scientists, okay? If we can interpret or explain this in a more common language, I think it will allow the society to understand the potential. And it will mobilize the government and also the private sectors to put more fund for this investment. I can see the benefit. Really, it's not just a spinal cord. There are other parts that we can apply this kind of scientific knowledge. So I strongly recommend that putting some of this progress in plain language so we can understand and society can be mobilized to support this one.
Thank you for your great summary. We have time running out, right? Therefore, we would like to discuss more. By the way, let me close this moment. Sorry, Peter. Yeah, our organizer said, let's close. So it is fine. Yeah, thank you for giving us your great science, Professor Nakamura, Peter, and CEO and the chairman of Nomura CIEA. Thank you so much.
Thank you very much. Sorry to rush you in a very wonderful discussion. Now, this is the end of the session 2, and I would like to invite Mr. Yoshiyuki Makino, Director of Science and Technology Innovation of the Kanagawa Prefecture, for the closing remarks.
I cannot speak English. Sorry. はい本日は、夜の殿町での熱い時間の共有ありがとうございます。そんな笑うとこじゃない言いますか。佐藤じゃ訳して。
はい、firstly, I'd like to thank all of you for sharing your passionate time with us tonight in 殿町。
今、日本は、失われた30年と言われる中で生みの苦しみの真っ最中でございます。
It is said that Japan has experienced the lost 30 years, and we are currently in the midst of pains of social and economic changes.
で今日のコロナからの科学技術と社会の対話等の教訓、そして再生細胞医療と異分野融合という希望の光、こういった観点を軸に、日本から世界に貢献していくイノベーションを創出育成していきたいと考えています。
Based on the lessons we gained from the COVID-19 pandemic, which is the importance of dialogue between science, technology, and the society, as well as the hope of regenerative medicine and interdisciplinary research and development, we wish to create the foster innovations in Japan that will contribute to the world.
この殿町をですね、基軸にスタンフォード等々グローバル展開も進めていきたいと考えていますので、スタンフォードの皆さん、今後も明日の未病サミット、こちらの方もどうぞよろしくお願いいたします。
We would like to promote global collaboration with Stanford University and using this Tonomachi as a foundation. And we are looking forward to working with you all. And we also hope we will have fruitful discussions at the Mibyo Summit starting tomorrow in Hakone.
でそしたら最後にですね、日本の今日の登壇者の皆様と私たちが日々共有しているですね、日本のイノベーションの極意を共有したいと思います。
In closing, I would like to share with you the essence of Japanese innovation that I share with all of the Japanese today's speakers on daily basis.
私たちの合言葉。
Our motto is
辛抱、所持、貫徹。
Probably in English it'll be be patient, follow through, and accomplish what we set out to do, what we set out to achieve.
あさっての未病サミットで決め言葉もこれになりますので、スタンフォードの方もリピート、OK？ OK？ じゃあ、OK？ じゃあ皆さんも合唱していただいて。言ってくださいね。はい、じゃあせーの、はい。辛抱、所持、貫徹。以上でございます。今日はありがとうございました。
Thank you very much for your time today.